Let me introduce myself. I'm Jonathan Pollock, and I'm the chief of the Genetics and Molecular Neurobiology Research Branch within one of the divisions of NIDA in the extramural world. He basically runs the intramural research program, which is the government hires its own scientists, but most of the funds for NIH are extramural funds, as I will show in a minute. And there's also another division, so I'm in the division of basic neuroscience and behavioral research. There are other divisions as well, such as Division of Epidemiology, Services, Prevention Research. There's another division which does medication development and funding, and also there are other entities within the Office of the Director, which does everything from communication, etc. So first, what I'd like to just cover is, first of all, I want to how many of you have applied for a fellowship or a grant? So how many of you have actually interacted with people like myself who program? So good. Now, what I want to make the point, and clear, is that whenever you apply or you're thinking about applying, it's always good to talk to us before you apply, because we can give you advice and we can also tell you what we're interested in, okay, which may help you later. And in terms of the process, and I'll just give you a quick thing for the process, so is you submit an application, it's assigned to an institute, and you can request which institute to be assigned, but it should be relevant to the institute's mission, because we're mission oriented. And second, it'll get assigned to a study section for review that has the expertise. And then there are typically 3 people who will review this in a study section, and then they'll give their initial level of enthusiasm. They'll discuss why they think the score is appropriate. The score is usually 1 to 9. They'll take the top. They'll usually only discuss the top half of the applications, but everybody gets a summary statement back. And then what happens is it comes back to program, and program at my level. And first we have a discussion within our own branch, and based on the scientific merit, the availability of funds that we have, and programmatic need, our interest, we make recommendations to the division director, and we meet with the other branch chiefs, because there are other branches within my division that do more stuff related to electrophysiology or more behavioral pharmacology, etc., or circuitry. And then we'll get together, and then we'll intercalate and make sure how that gets done. And there's a certain formula actually for it, but I can go into that later. And then we may come and we meet with eventually council has to basically concur with the IRG recommendation. So basically that is done en bloc. And sometimes council will make a recommendation that one should fund this versus something else. But typically it is just en bloc. And everything really is a recommendation. So the ultimate power to fund or not fund rests with the institute director. That's the only person, except for grants management, that can actually fund something. So that's what you should know. So recommendation is made, and we all meet with the institute director. So you can have a worse score, but it has high programmatic relevance, so that might get advanced over another thing. So it always pays to talk to program. So and don't be scared of us. We're here to help you and give you feedback. So, and to advance the best science for the American taxpayer and for the world. So that I highly encourage you always to talk to us, and don't be scared of talking to us. Okay. So that's the first point. But I just wanted to go and talk briefly about the NIH budget. So the budget is about $30 billion, which is 3/5 of what the State Department's budget is of $51 billion. And most of the budget is in research projects grants. These are the RO1s and PO1s, and the other, you can see a much smaller fraction is in the intramural research program, which accounts for 11%, and some of it is in contracts, which is another 10%. A smaller portion is in research training, and other research grants are like R13s that fund this meeting, and then there are research centers like the P30s and P50s. Chris Evans has a P50, and also Angus has a P, along with at Yale has a center for proteomics. So that's a much smaller portion, but they're separate portions of the budget. So if you look at the NIH budget in terms of constant NIH dollars, it steadily increased over time, and then from 1997 to about 2003, there was a doubling, and then it has slowly decreased. So if you look at the rate at which this grew, it grew approximately at the same rate as the US GDP, but it's now declined. So if you look at the success rate, I think it's in the next slide. Oh, I just want to, so this gives an idea of how the NIH budget is sort of divided up. So here's the NIDA budget, which is $1 billion, and the NIMH budget is about $1.4 billion. So the largest institutes are NCI, which actually is in a sense a separate entity from NIH because it's the only institute director within NIH that has to get a Senate appointment, other than the NIH director. But that has to do with history. And so the other large institutes are NHLBI, and NIAID, and NIGMS. So if you're looking at the success rate over time, you can see that the success rate for NIDA new applications and NIH applications has basically declined since 2001. Okay, but what you see is that more or less that NIDA and NIH are, it's more or less the same if I've got this right. Okay, but what I'm trying to say is that if you are a new, if you're submitting a new R01 application, your chance here is about 15%. Whereas if you're submitting a Type 2, a new competing, a competing application that you had already been funded previously, you have twice the rate of success. But what's really happened over time is that the number of actually awards is pretty much stayed pretty much constant. But what has also happened is that the number of applicants, and I guess you could also argue the population has greatly grown, so that the success rate has somewhat fallen since 2002. And the good news is, even with sequestration, okay, even though there's been a cut, what hopefully will happen, at least in the president's proposed budget for FY 2014, which is the coming fiscal year, and the fiscal year begins for the federal government October 1. It will restore the cuts. So I just wanted to give a brief overview, and I don't want to go into too much, keep you from dinner and other things.
It's changed. It's all changed.
It's all changed?
They screwed up our schedule, so we got regular dinner. And we're gonna the wine and cheese now is 6:30 at Roberts.
Oh, okay. All right.
So
my program and what I'm interested in is basically, human genetics, model genetic organisms, epigenetics. I also support genetics of modulation of host response for HIV, pharmacogenomic response, and also brain molecular genetics of brain development. Oops. Oh, I guess I got the slides a little mixed up. But this just basically shows the portfolio that the genetics at NIDA has. So you can see that fair amount is into molecular pharmacology, about 14 million of the total R01 budget of about $60 million for genetics. But the total, genetics budget with including other divisions for epidemiology, et cetera, is about $118 million. Okay, so just some of the highlights of what we've supported in the past, is, for example, we've shown for coffee consumption is associated with CYP1A1 and 1A2. Also, one of our big discoveries that we've supported is the whole, 15q25 with alpha 3, alpha 5, and beta 4. Also, in terms of smoking behavior for CYP2A6, and also a clearance of HCV, we've also supported that as well, which turns out to be this gene called IL-28B. So this is just what I think David Goldman previously showed for all of the success is for GWAS. And there is some evidence to suggest that neuregulin 1 is associated with cannabis dependence, which is work by Joel Galertner. And I think you've seen this earlier today, just showing that, work by Paul Kenny basically showed experimentally that you could get the same phenotype as increased number of cigarettes smoked in mice. And then I think this was alluded to earlier by Marina, that basically the variant for alpha 5 decreases the permeability to calcium, as can be seen here. And that's work by Jerry Stitzel that we've supported. And then also in that cluster of, I'm talking about drug development, the alpha 3, beta 4, there's also work that Larry Toll and Neuroline Severi have developed on alpha 3, beta 4, where there's an antagonist, and this decreases in a dose response the amount of nicotine that's taken, but doesn't affect food. And you can also see that the high risk variant has relevance for treatment, that people with the high risk variant show a greater response, or more likely to respond to nicotine replacement than those people who don't have that variant. So, and I want to, so in terms of model genetic organisms, this is work by Abraham Palmer's group, and he's done mapping in terms of the locomotor response to amphetamine and fentanyl, and he shows that casein kinase 1 epsilon is associated with the locomotor response. So you can also do looking at recombinant inbred animals. So you take one strain, say black 6 versus DBA, and then you can do, you basically can cross them over a number of generations and develop recombinant inbred strains. So this one is work by Mike Miles and Imad Damaj, where they basically show that that if you're looking at withdrawal, the withdrawal phenotype is associated with the galanon receptor GAL1. So coming back to the issue of impulsivity and mapping, which I'm very interested in terms of inbred strains, is the issue of novelty seeking and self-administration. So if you put an animal into a chamber Initially, they will just locomote around, and then they'll habituate. But if you have, and that seems to be associated with the amount that an animal will self-administer, but it's not, well, I'm not saying that right. I'm saying is that if you allow the animal to then subsequently seek to go outside the box, those animals that show greater novelty preference of seeking outside the box after they've habituated show greater self-administration of amphetamine. So there are looking at phenotypes that are associated with addiction may be better models for looking at the genetics of addiction in rodents. So I'm very interested in, would love to see applications that really map this type of behavior. And then again, there are some other phenotypes, as I said, novelty preference, but just not locomotor reactivity, predicts the development of, cocaine addictive behavior. So I think you'll hear about this tomorrow from Bellin. And then also high responders, initially predict the propensity to acquire cocaine self-administration. And then again, the issue of impulsivity predicts, increased compulsivity. So these are types of behaviors that I would be very interested in using mapping strategies in rodents to, identify new gene variants that we could subsequently test in humans. And also work by David Jensch, looking at reversal learning, which seems to be a good model for impulsivity, suggested that these genes, SYN3, NT5DC, and 8CFC2, which I don't know exactly what they are, does seem to be associated with impulsivity and potentially with substance abuse.
He thinks it's synapsin 3.
Yeah, synapsin 3, right.
That's what.
And, one of the, in terms of mapping strategies, there is a development of what's called the collaborative cross. So this is there are 8 parental strains, and what they've done is they've basically taken the different parental strains. So you can see that in the first generation, you basically get the one chromosome from each parent, so they're basically heterozygous. But then what happens is in the next generation, when you do the cross here, you get recombination because you get crossing over. And so by subsequent breeding from the same brother-sister matings, you can fix the genotypes and you can do mating. But also what you can do is you can also jumble these up in a way so that you basically get highly diverse group of animals so that you can do actually a genome-wide association scan. So it's possible now, say, for example, what David Goldman was talking about, 5-HT1B receptor knockout in terms of impulsivity, you could potentially look for dominant mutations that would act as a modifier for this. And you can do this in about 200 to 300 animals. And so the other interest that I have, and I won't belabor it, is where we've supported a lot of work on the epigenetic modifications and drugs of abuse, looking particularly at chromatin modifications, stuff on delta phos B. So, and tomorrow I think you'll hear about work that my program supported of Chris Pierce, basically showing that if you expose in the previous generation cocaine, they actually show less susceptibility to the locomotor response to cocaine. So just to give you an idea, we're very much interested in identifying biomarkers and identifying rare variants using deep sequencing approaches, looking for it in population isolates, to identify basically the mechanistic roles and the functional roles of the genetic variants that are discovered in human populations, as well as going back from rodents to show that it does exist in humans, and to really leverage and begin to leverage bioinformatics and computational approaches that integrate existing data sets and really use big data to basically identify new variants and to find and to assess their function. And then in terms of model genetic organisms, as much as I'm a big fan of knockouts and push the development of the knockout mouse project, I think I see great opportunity right now in terms of mapping natural variants and also doing selected breeding, as David Goldman discussed, and really identifying those variants and really looking at individual differences in vulnerability, such as impulsivity, stress reactivity, not necessarily looking directly at reward. I think what I heard, been hearing today and yesterday, is that you really see that the field is moving to what drives sort of compulsivity, and it's not directly the reward pathway, but rather it's other systems such as the prefrontal cortex and other areas as well, or as Paul Kenny talked about, area X in the thalamus. And really we can also exploit some of the mouse genetics really to look at toxicology in terms of drug development. So for example, rimonabant looked like a pretty good drug initially for treating smoking as well as obesity, but it had all these side effects. So you can use perhaps the diversity outcross that I was telling you about, where you do a GWAS and looking for adverse events to identify those variants that might be associated with increased anxiety or increased or depressive-like behavior in mice, and then test to see if that variant exists in humans. So that could be potentially useful. And so we're also, as David Goldman talked about, there are somatic mutations within single cells. We're very interested in how single cells play, basically being able to analyze single cells, and also optical genetics, and really mapping, developing research to look at cell identity, connectivity, and variations of cognitive and behavioral circuitry as it relates to genetics. And I think there's going to be a big opportunity eventually in terms of being able to barcode cells, in terms of their assigning basically a DNA barcode to cells, so that you can individually profile a single cell and look at its connectivity with a DNA barcode. And then identifying genetic variants that regulate, variation in neuronal circuitry. So if you are really able to identify and DNA barcode individual cells and their connectivity, then being able to look at individual variation in neuronal circuitry will become quite possible. And expensive. But and also, I think there are now great opportunities with zinc fingers and TALENs and the CRISPR to really directly modify any genome from rodent to primate. And so
we could get William Yang to talk about the talent.
Right.
He knows about that.
And so, another large portion, which is run by John Satterley is our epigenetics program and non-coding RNAs. And we're also interested in, DNA binding proteins as they relate to substance abuse. So we're very interested in the role that non-coding RNA regulation in neuroplasticity and addictive processes, and a careful and systematic exploration of RNA modifications in the nervous system. We like shRNAs. We're very interested in long non-coding RNAs. We like to create a comprehensive atlas of molecular phenotypes for specific cell types in both human and mouse, which is some work that we're right now supporting by Nat Hines, who just was awarded a P30 at Rockefeller. So he's going to basically use the back traps to profile different cell types that are associated with addiction. And there's also work going on at IRP along the same lines, and to really develop small molecular epigenetic modulators that target epigenetic modifications in addition to, using, say, talons that could basically, modify, you can use it to modify epigenetics if you put the right enzyme along with a talon. And examine sort of some of the stability and dynamics of somatic epigenomic changes in response to drugs of abuse. So that just gives you, I'm giving you a flavor here. And we're also interested in pharmacogenomics. So using, for example, looking at alpha-5 and seeing whether you can basically predict, based on alpha-5, some treatment response to various smoking cessation agents, and also CYP2A6. And hopefully we can also do this for opiates as well as other medications to basically predict what would be the most effective treatment. But we're not quite there yet. But it would be useful in terms of, for example, predicting who might respond to naltrexone therapy for A118G, which seems like males seem to respond, alcoholics seem to respond to it, have that variant in the mu opiate receptor. Okay, let's see. So also, we do also, because HIV and HCV is associated with addiction, and actually, in some ways, it's a good marker for addiction. We are interested in looking at the host response. So looking at various systems biology, epigenetics, and biomarkers of HIV progression, and we've had RFAs and PAs in this area. So, for NIDA, about 1/3 of our budget is dedicated to HIV research. What's interesting is that in terms of public health right now in the United States, HCV will be a larger problem than HIV in terms of healthcare costs because of the cost of transplanting livers, and it's sort of like this crisis that will happen, in 10 to 20 years. And also the thing that if you know people that are have small companies, we're very interested in building the SBIR program. The SBIR program, it accounts for about 5% of the NIDA budget. It's dedicated by Congress for the purpose of developing small businesses and translating, basic research findings into something that becomes a product. And so what's happened, one big change now for the SBIR program is that before, if there was any venture capital money invested in a company, they're not eligible. And a small business is anything under 500 employees. So anyway, but just to give you an idea, some of the things that we're interested in is protein capture, so antibodies, gene delivery, analysis of trying to basically profile fixed tissue, which would be, very useful for looking at postmortem, a lot of postmortem tissue that's been archived for years and years. With, if you can actually do gene expression, that may be very useful. IPS cells for therapeutic development, and various methods for doing data analysis, integration, and visualization. So I just will stop right there and open to questions and your concerns. And please, if you have any questions, call us. We're here for you.
Your investigator strategy, that was a big one.
Okay.
So maybe some questions on
so
how they would go about getting book grants or available.
okay, so typically if you're available for graduate students, are existing training grants that you get onto, which are basically initiated by the institution. Or there's an F31 mechanism for graduates, for graduate students, so that you can write for that. There's also, I forget the exact mechanism, and I'd have to look it up. There is a dissertation, there's a special mechanism for dissertation research. In terms of, when you become, as a postdoc, there's opportunities to write for it's a Ruth Kirstein F32 award. So that goes for 3 years. And you can also be, supported by the investigator that whose lab you're in. And then if it's always, so when you apply for these the things, they're more focused on training. So what's the training program? What's the quality of the candidate, and less so of what the research project is. But it should be a solid research project that the review committee sees as doable. Also, the other things that need to be worked out in terms of training is evaluation of contact time with the person who is your mentor, conduct, ethical conduct in research. And I think also an important thing would be to have courses in research design, because a big concern right now is that a lot of stuff that is published can't be replicated later by pharmaceutical companies. So you would expect with P less than 0.05 that 1 in 20 1 in 20 papers you would not be able to replicate it. I don't know, but you have non-replication now in 50% of a lot of papers. So research design.
50%?
Yeah.
What?
Yep. It's high. And so learning how to really write a method section so that people can replicate the results that you did. The method section probably is the most important part of the paper. So that's one thing. So at the end of the F32, you can either go out, and look for a job, or if you want more training, there is the opportunity, but you got to go do something somewhat different, in another lab. There is the opportunity for a career development award, a KO1, or if you're just a PhD. If you're just an MD, you can get what's called a KO8. And then for physicians, there are a lot of clinical training mechanisms as well. For other mechanisms for training, if you are a chemist or a computer scientist, you can, and already have a PhD, there is what's called a K25, which basically pays a salary equivalent to a congressman. There's, let's see, what else can I tell you about that? The K99. So if you've been in a lab as a postdoc for a while, and I forget the exact, 5 years.
They just
With
produce
5 years.
Is it 4?
It's now 4?
Starting February of next year, it's 4 years.
Okay.
Yeah, just changed it.
So if you've been in 4 years, there is,
Pregnancy counts as you can take that off.
I mean, it's not long enough. A lot of these projects now.
They take that long.
We
Like,
take
Yeah.
that.
Why
it takes that long.
did they reduce it to?
I don't know. You see, I think, I'll tell you what the problem is that there's real concern at NIH that most PIs are now becoming PIs at age greater than 40. So there's an,
So you're going to reduce the time to try and get us off faster, but it doesn't work.
well, there's also effort there for people who have just gotten their PhDs, who are really, stellar, the opportunity to basically there's a startup grant for that as well.
So maybe do a 7-year PhD and do all your postdoc work in your PhD.
I mean,
I don't know what to think about. I mean, it seems like people evolve at different times. There should be opportunities at different times. Right?
well, I'm just telling you what, I'm not on the training committee, but, so for the K99, it's basically 2 years, or 3 years, I'm sorry, 3 years, and then basically if you find a faculty position and a way to transition, you have 2 years as an R00.
I think it's 2. I think it's lower than that. It's 2 years. So you've got
Yeah.
one, right?
It's 2 years plus a no-cost extension
Right.
if you need it, and then you get
Right, okay.
3 years of the R01. But you have to find a job
You have to find a job.
in a different place. Now, is this open for non-residents and residents?
I need to double check on that.
K-99 is.
I thought
Yes,
it was.
the K999.
Yeah, it's open to non-US citizens.
Yes, yeah, that's what I figured.
Right.
That's one of the only grants which is.
Right,
The
well,
NRSAs.
all the NRSAs are all national, you must be a US resident or,
The statistics I saw for MDs on when they get their first R1, actually the average age was 46, which is basically my age. Can you imagine? That's ridiculous. So it's a huge problem. And one measure that medical schools are taking all over is that they combine MD PhDs into shorter programs, into some sort of highways to make sure that you guys don't end up being, my age and getting your first R1. So that's 46, I think, average 46.
yeah, it's,
Maybe there's some
and
who just
even
don't
for,
get any.
but even for PhDs, you see like 40.
Yeah,
42.
Yeah.
So, I mean,
So you see these guys know all the stats better than you. I mean, it means they've been looking. I mean,
good.
they're worried about you.
I know they're worried about it, so, well, what can I tell you? What more?
I have a question about the postdoc funding.
Uh-huh.
So I had a discussion about this with people earlier. If your advisor has a grant,
Right,
okay, on a certain project, and that is your project, is there any problem with you applying for your own grant as a postdoctoral training fellowship for the same project?
I don't think so.
As long as you write it yourself.
Sure.
So it's not considered double dipping.
No,
I don't think so.
No.
One's a training grant, the other's like a research grant.
No, I think it's okay.
it's fine.
It's fine. It's absolutely fine.
So it can't be the same project name, right? It can be identical.
Well, it shouldn't be, obviously not identical, but if you're part of the
A
R01,
twist, yeah.
yeah,
If
because
what you're
probably
doing
your
is
postdoc
supported by
project isn't
another
the
grant,
whole
doesn't
R01.
mean that you can't write in an RSA.
Yeah, but if you are part of an R1,
Right.
technically that grant that you're submitted has not been awarded yet. You as a postdoc can write 10 R1s a year if you like, right?
If the institution allows you to write an R01.
Exactly. Most institutions do, like UC, etc. But then, if you get the money, now that's a different story. So until you don't have it, it's kind of an Epicurean.
Well, let's say we have-
He's talking
No,
about-
but he's talking about an NRSA. He's talking about a F32.
Line of work,
That should be
and your
okay.
component of that project.
Yeah,
or-
you
Because
can,
it's a training grant.
it's meant for training.
But you
So-
still get
so
critiqued for
Jonathan.
being too
Yes.
similar to your PI.
Or do you?
Are there
Do.
grants that are postdocs or scientists certain names that you know that stuff I don't.
No, I
Is
mean,
F's, U, something that is not taken advantage of.
the UO1 is a cooperative agreement, but
Yeah,
that's something that has to be really initiated by program.
Do you know about this K-18? It's a Career Enhancement Award. So it's supposed to facilitate translational research. So if you're in a basic science lab, you can write a grant to do a small project to go to like a human clinical lab,
Mm-hmm.
or vice versa. But I'm not sure, like, I mean, I'm finishing my 5th year as a postdoc. I don't know if it's a good idea for me to extend, to apply for that type of grant. You know, who is this appropriate for? And what, you know, is there a good time to
When
apply
I heard
for
it, it
that?
seemed like it was for like a younger postdoc. And it has to be at an institution like far away from your own. Like it
Really?
separate from your
I
own.
didn't
Because I was gonna
Any-
apply for within my institution.
Yeah, any of the Ks after you've done the F-32 should be on something different, in a different direction that you've been going. But you also have to say, how will it support where you're going to go ultimately? I mean, the problem is you change direction, then it takes a long time for you to gear up again to be productive.
So what percentage of grants are ones that are being funded by NIDA at the moment?
Well, I just showed it's about
Oh, sorry, I missed the part, first part.
It's about 15%. It's about 17%. You know, and if you are competing, you know, for this application that you had, it's somewhere around 25 to 30%.
That's much more than I thought.
I mean,
For all of the versions of the grant, total.
So,
That's from the first time that you, A0.
if you go here,
There's only 2.
Yeah, it's not all 2.
Only 2.
All 2.
But that's higher than a lot of places, which, I mean, NIH is going to like 5.
Yeah.
So
well, it was
Just pass this out.
passed it. So if you go here, okay. So this is purple is NIH competing, NIDA competing. So NIDA is a little less, you know, in terms of competing. I guess one is 35 versus maybe 25. Okay, but if you look at, you know, new competing grants, we're exactly the same place. Oh, let's see, blue NIDA new and red NIH new. We're in pretty much the same place. In fact, we're in 2012 we were just a little bit better.
So these are our ones to the training awards at Cellular?
I don't have the data for the training awards.
They vary by year. Like, not like a wall.
What about those K, like KO5, KO1?
Oh, okay. So KO1s are given if you have some type of independent research support. So that's for KO1. Well, I'm sorry, KO1. I'm sorry, I'm talking about KO2. But KO1 is a mentored research award. So after you've done an F-32 and you're looking for another avenue for training, and it's something related, you should do something related, but explain how this training will ultimately benefit you. And the institution should provide some means by which you will be able to write an R01 from that KO1. That's one. Now, in terms of KO2, is you already have research support, right? And you write for a KO2, which is meant to basically support more career development and training and free you from some of the administrative and teaching burdens that might be associated with a job. So NIDA supports the KO2 right now, but is basically phasing out the KO5 for senior investigators.
What about R21s?
R21s are meant to be exploratory and innovative, and I would discourage new investigators from doing it because it's, I think, for two reasons. One, it's short-term funding. Secondly, you know, it's based supposedly if you're doing something high in risk that's high risk, people are supposed to judge it on your past track record. So, you know, and I think institutions for tenure look at whether you get an R01 or not.
And if you have an R21 awarded as your first grant, you lose your new investigator
Yeah.
status.
That
right?
too.
That's
Mm.
true.
So you're, yeah, that's important. So you're basically burning your,
Yeah.
you know.
But at K99, you're still good. for
Right.
the first,
Yes,
Which is.
yes,
Right, you lose, that's right, you lose your new investigator status,
exactly.
which helps you. So that, so if you have never had an NIH grant, you can be a new investigator. The other thing you should know about being new investigator is if there are minor criticisms in your application, and you can fix it, but you really should talk to the program before you do it. You can come back in the next round instead of waiting 2 rounds to reapply. So
And are there still higher chances of your R01 being funded if you are a new investigator?
Yes.
in
Yes.
general?
So, I mean, there's also a study section, they're identified as being a new investigator, and so it can influence the way the discussion goes about minor
and then there's,
problems associated with components of the grant.
So it just means you've never had an R1?
yeah, and then I forget the other category, which is if you've been within
Young
5 years.
investigator,
Young
yeah.
investigator also helps too. I mean, I have recently there was a situation where there was a pretty senior investigator who never had an R01, was classified as a new investigator, which I thought was kind of funny, but because of that, and there was some criticism in that he could rapidly address, he was able to put it into the next round, and it went from basically a really crappy score to the best in the pile.
So Jonathan, imagine that one of them gets a job tomorrow, right? They become assistant professor. What is the sequence of grants that they should apply to? R01 first.
I would then also advise them to look at all the opportunities for various foundations.
But from NIH, I mean, what kind of
I-
portfolio should they be? R1 plus?
I think they apply for an R01 and then apply for a KO2.
Okay. Those are the 2 first things you would.
Right, and so, but the one thing that sometimes happens is if you have an R01, they may, and then you ask for salary support for your KO2, they may take, if you had already requested salary support on your R01, they will take they'll take money away from that R01. So if you have support from your startup funds in the first 3 years, right, and you apply for an R01, don't put your salary on it, and then apply for the KO2. But the downside for that is when you eventually come back and you're no longer eligible for that KO2, it will start to come out of your R01. So you reap the benefit at the beginning, and then you may reap the pain at the end. And I think Mark Benzaster can tell you stories about that.
One thing that's really important to remember with a KO2 is that it is for 75% of your salary, and there is a cap on funds of $90,000. So if you do not have a source for 25% of money that is not federal funds, can't be your R01, can't be an R21, can't be an R03, has to be a completely non-federal source, so it could be funds from your university, it could be funds from private foundations. That 25% has to come from those non-federal monies. And so you can find yourself at a soft money institution regretting your KO2 sometimes.
So you
You
should
have
make
to know
that
that
deal
you
when
have-
you make the, you should make that deal when you,
That's correct, yeah,
when you become assistant professors.
yeah. Or when you apply for the grant,
Or when you apply for a grant.
yeah.
So one of the things that Jonathan had on his slides, which is useful, was the program announcements, the PAs and the RFAs. You know, it's useful to get on the listserv that feeds that information and tells you
Right.
what's coming up.
So every Friday, so you go to search NIH Guide, and every Friday are new announcements that come out in the guide, and you can register to be on the guide. And sometimes what I do to highlight what my division is interested in is I will send out a mass email to 60,000 of my closest friends. It's called the NIDA Neuroscience Update. So it's the NIDA Neuroscience listserv. So, I mean, keep an eye on the guide. There's always something, there's always something that's happening.
Guys, one thing I want to say, because I'm basically an electric neural animal that just moved to NIH. So one thing that I finally understand seeing Jonathan and all the people in the administrative side of NIDA, he means what he said. They need to find and connect with the best and the brightest scientists in order to get them into the NIDA portfolio. So it's not that NIDA on one side is trying not to give you money and you are fighting to get the money. He wants all of you to succeed and get basically money from NIDA. It's in his best interest because it's in the best interest of NIDA. So there's no conflict. When he was telling you that he wants you to contact him to apply, to ask him questions, it's because he needs to get, ideally, the best possible scientist to be sponsored by NIDA and to be funded by his program. So there is communion of interest. It's not
Right. And
don't
also,
think for a second that one is going in one direction and instead there is a bunch of people there trying to not give you money. It's
Or
the opposite. Right?
right. And also, we know a lot of stuff that's going on because we read all the grant applications. We attend the study sections and we hear stuff. So we may not be able to tell you what somebody is doing, but we may tell you, go talk to this person because I think he has something that you might be interested in. And that's a big hint, So, I play matchmaker for various interests, saying, you should go talk to so-and-so because I think there's something of mutual interest. And I might not be able to tell you what it is because it's confidential. But the investigator can, if the investigator is interested in talking to you, and you say, Pollock said, come talk to you because I'm working on this project, and he said there's something that you're doing that's of interest. It may be a little uncomfortable, but I would encourage doing it. Or you say, or sometimes I'll just get on the phone and I'll say, hey, I can set up a conference call between the two of you or the three of you to basically get something going. So, I mean, that's another reason to do it. Now, one of the problems that we're having right now is in terms of travel. It's the number of people that we can travel to, say, Society for Neuroscience is limited. So you can't talk to all the program staff at one meeting. A lot of the stuff has to be done by phone, but, we have if you want to see us, we have ways, through web communication that you can see us and talk to us. If you want us to basically have a seminar, talk about funding at your institution, I can set that up by phone and by WebEx. So if you want, want more information about this, and you go back to your institutions, we're happy to set up, a video conference. And you can, and also, it's, if you want us to come out and site visit, that's easier in some ways than going to a meeting, although we still have to justify it.
So looking at this graph, it seems like things are definitely stacked against new R01s. Is that because it's a safer bet for NIDA to fund the competing ones, or are they less money, or what is the rationale?
Well, one, you've already gone through the system one time and you're successful. The second thing that's happened is you've accumulated a whole number of more data and papers on what you're doing. So you may, and frequently investigators will change somewhat of the direction that they're going in the next application. But there is a certain amount of confidence. Also, in terms of the funding, if I see a type 2 and it's an A1, so you've got 2 shots. But I'll come back to this in a second. I will frequently go like, well, this is a continuation of a program. I want it to continue, so I'll make the recommendation that it should get funded, even though it's a worse score, because what's going to happen is that program will just completely end, at least with that mechanism. So if, say you go twice with an R01 for an idea, so you go O1, A1, okay? You can't submit the same thing again as an R01 as an A2. But what you can do is you can submit it with another mechanism. So you can submit it as an R21. You can submit it as part of a program project. You can submit it as part of a center with the same specific aims. With an R21, you'd have to cut the budget and probably cut 1 or 2 aims out of it. But you can basically do it again. So that's what I can tell
Or
you.
you can sneakily change the name and change all of the abstract.
And the aims.
Which I think has happened before.
Yeah, I'm sure that's happened, but
Yeah, but
I wouldn't-
the study sections get pissed off about that from what I understand.
and program also notices it.
Yeah. Yeah, I'm sure. So it's something to avoid.
Are there any mechanisms that NIDA doesn't fund? You know, sometimes there's.
Oh yeah, different is you have to look at, for example, there's a resource thing like an R24. I mean, you can go and we don't fund it, but say NIAAA funds it. And what I use for that type of thing is a P30 core in its place. But if you ever go through and look at mechanisms, if you search mechanisms, funding mechanisms on the web, there are a huge number of mechanisms that are there that are not necessarily used. And then there are these Pioneer Awards for new investigators and Innovator Awards and the Eureka Grants, but they're competitive.
Well,
Okay, it should be fast. So in terms of, we were just talking about funds, but like other resources. So earlier I kind of mentioned the NIDA drug supply program, and it's like a collection of small molecules that NIDA investigators have access to for free if they can justify. Is there anything else like that that we should be aware of?
There are large numbers of resources that you can search. The Neuroscience Information Framework is like one-stop shopping for research resources. You can also go to the NCBI page, and it lists a lot of, particularly genomic resources.
But nothing else specific to NIDA that would be like that.
I mean, there is like the NIDA Genetics Consortium, which has, we have 50,000 cell lines associated with, addiction.
Are we on a NIDA Neuroscience listserv?
Send me an email, and I'll give you the email address. It's
maybe you can put everybody on. Yeah.
I don't. Yeah, if you give me the list of emails, I can. Yeah, I can
If
put.
that's all right, does everyone want to be on it? Yeah. Lots of nods. Wine and cheese.
Wine and cheese. So I'm here till tomorrow at 5:30, and I'm happy to entertain any questions, ideas, etc. I'd like to talk to all of you.